Good morning, everyone, and welcome to today's MPG Primer. We are very excited to have Dr. Katherine Chao here today. She's a software product manager in the Translational Genomics Group, where she works on this little database you may have heard of called the Genome Aggregation Database, or gnomAD. She's the product manager for gnomAD, leading the production of gnomAD releases and managing the roadmap for the future. We've all been very excited about the new availability of gnomAD v4, and so we get to get a little bit of a behind-the-scenes tour today, as well as an update on the new features. So thank you so much for joining us today, Katherine. She's happy to take your questions during the talk, so please just come up to one of the mics if you're in the room, or post in the Q&A if you're online, and we'll voice your question for you. Thanks so much.
Thank you. Thank you for giving me the opportunity to represent our team. And good morning. Thank you for coming so early to my talk. So, like Sarah said, I am the product manager of gnomAD, and today I'll be giving you a glimpse into how we created the latest release. So I'll go through four major points in this talk. The first one will be why gnomAD. So why do we care about gnomAD, and why did we create a database like this? In the second section, I'll talk about how we actually created the gnomAD release, so version 4, and this will be an abbreviated overview because obviously it takes more time than I have to talk about how we make this database. In the third section, I'll talk about what's actually in the newest release, so version 4, and then I'll wrap up with a few of the most frequently asked questions our team has received since the release. So moving into part 1, or why. So to understand the variation within a single individual, you really need to understand how frequently that variation occurs across the human population. And to put this guiding principle into context, there's a really motivating story for why we created the genome aggregation database. So nearly a decade ago, Heidi's lab detected a variant in PTPN11 in a fetus with increased nuchal translucency. That particular variant had been observed in a patient with Noonan syndrome and was reported as pathogenic at the time by a well-respected lab. So to try to understand how frequently this variant occurs, Heidi tried to access a research data set with individuals with matched genetic ancestry to understand how frequently this variant occurs in that group. Unfortunately, she was denied. So based on the information available at the time, Heidi's lab reported this variant as likely pathogenic, and the couple terminated their pregnancy. Later on, they discovered that this variant was actually common in this genetic ancestry group and was benign. So another quick story is, in 2016, a group looked at variants that were considered disease-causing or pathogenic for hypertrophic cardiomyopathy. And they identified variants that were considered likely causal, but were actually common. So they found that patients were misdiagnosed due to a lack of access to allele frequencing data from diverse individuals. So how might both of these stories have ended differently? There might have been a different outcome if there was a reference allele database that had enough samples to ensure accurate frequency estimates, that included globally diverse samples representative of the human population, and importantly, that was free and had open access to researchers. So on the gnomAD team, we firmly believe that for the best outcome for all patients and to understand the impact of human genetic variation, we really need to enable data sharing on a global scale. And so with that, we created the genome aggregation database to kind of check off these boxes. So gnomAD is a frequency database that has enough samples to ensure accurate frequency estimations, that has globally diverse samples, but I will note that the project, like the field as a whole, has room to improve in this space. And importantly, gnomAD is free and open access, not just to researchers, but to anyone that has internet access. And quickly, who is in gnomAD? So gnomAD is a database that contains sequencing data primarily from case control studies of adult onset complex disease. We have two data types, exomes and genomes. There's a rough 50-50 split between samples that have inferred XX and XY sex karyotypes, and the mean age is about 55. So moving into part 2, how did we actually create gnomAD v4? So I know this is very overwhelming as a workflow, but this is the workflow for version 4, and you'll see that there are 3 streams here. So the first one is for the short variant call set generation, and this includes calls from both the exomes and the genomes. We also have a pipeline here in the middle for copy number variants, and these were only called on the exomes. And then finally, we have a pipeline for the structural variants, and these were only called on the genomes. For the sake of time, I'll be focusing on the short variant pipeline today. And this workflow can be summarized into 4 major steps as well. So processing, data processing, joint calling, quality control or QC, and release. And I'll be going through these one by one, starting with data processing. So the gnomAD v4 database has over 800,000 samples. This plot here is showing the number of individuals in gnomAD colored by their data type. But this is the number of releasable samples that we were able to include in the final product. To create gnomAD v4, we actually had to process over 1 million samples. So this means that the genomic platform had to sequence all 1 million samples. Then project managers had to keep track of each sample and the mountains of metadata associated with the samples, including multiple compliance steps to make sure that we could actually include these samples in our data set. So once the genomics platform and the project managers have finished their portion, software engineers in the data sciences platform process all of the actual raw sequence data into genotype calls. Excuse me. Once we have genotype calls, the next step is to actually generate the call set or joint calling. So this is a particularly painful step at the scale of a database like gnomAD. So the traditional joint calling pipeline moves from individual level genomic VCFs or GVCFs into a single project-wide file called a project VCF. And it breaks down at scale. So to understand that, I have to quickly go through a project VCF. So this is what it looks like. It is a dense matrix. So this is a box matrix showing variance horizontally and samples vertically. And you can see that each box is colored in, which means that every sample has information at every variant site, even if they don't have a non-reference call at that site. If we had traditionally processed V4 in the same way, we would have produced a database that was 800 terabytes. So that is prohibitively expensive to compute on and just to store. So to build a database like gnomAD and to continue expanding the database, we really need scalable processing and quality control or QC. And this is where our team really leans heavily on the HAIL team from the Stanley Center. So to process V4, we used a data format from HAIL called the variant dataset. And to explain the VDS, we need to quickly go through a sample GVCF. So a genomic VCF or GVCF is a per sample file that contains 2 types of information. Variant site information, where a sample has a non-reference genotype call, and reference blocks, so groups of loci where the sample has a homozygous reference call at a certain level of confidence. Both of these types get directly translated into a VDS. So a variant dataset has 2 matrices within it, one reflecting all of the reference information and one reflecting all of the variant information. These matrices are sparse. So to show you what I mean, on the left side is a project VCF, and this is a dense matrix. Every box is colored in. But you'll see on the VDS, in the variant sites, for example, samples only have information at loci where they have non-reference calls. So you'll see that only this box in this first row, for example, is colored in. And in the reference section, samples only have information at the very starts of their reference blocks. They also contain information about the ends of the reference blocks, so you can infer how long a sample should have information. But you can see that there's a lot less information filled in the VDS. And this sparse data format is really efficient. So it takes more than 10 times less space to create a VDS than a project VCF. And this separation of reference information from variant information enables easier computation. So for example, if you have an analysis that only needs to look at the variant information, you can just read in this bottom matrix. You don't have to read in the entire data set to do your work. Also, importantly, the VDS is a lossless conversion from a genomic VCF. So every annotation that is present in a GVCF will be present in the VDS. And this isn't always true in the traditional pipeline. This allows for new QC methods because we just have more information. The VDS also really improves joint calling at scale. So it allows for N+1 processing. So this was a problem in the past where you had a data set with N samples and you wanted to add an additional sample. To do that in the traditional way, you'd have to reprocess the entire data set just to add that one person. With the VDS, you can simply add a GVCF right into the VDS. So the way that this looks, that arrow does not show up well. But so to make a VDS, you hierarchically merge GVCFs into a final data set. So if you don't have that many GVCFs, you can just merge them. That will be your VDS. In the case of gnomAD, we'll merge like maybe 100 at a time and then just continually merge until we have the final data set. And a variant data set, again, is lossless. So if you really need to, you can convert this VDS back into a GVCF. So this eliminates the need to store this separate data type. Yes, of course.
Hi, thanks. So I'm just trying to understand why the VDS is so sparse. Is that because there are 10 times more reference calls than variant calls in the data?
No, so it's the VDS is sparse just because it takes a GVCF and puts it directly into the VDS rather than trying to fill out information at each site, which is what you would traditionally do in a variant, like a project VCF. Because in a project VCF, you actually adjust sample genotypes at each site depending on what the other samples look like. So the call set could change based on sample composition. With the VDS, it's not truly joint calling because we don't adjust genotypes. All of the information directly goes from GVCF into VDS. So in a VDS scheme, no matter which samples are in your call set, the number of variants in a sample will not change, and their calls will not change in those variants.
I see. Okay.
Did that
But
answer?
it still means in the reference set, you're recording less information because you're just recording the runs of reference, not each individual reference point. Is that correct?
Yes, yes. So to convert from a VDS into a project VCF, you have to go through a process that we call densification, where you then fill out each site and then remove any sites that have only reference, as in a traditional VCF.
Thank you.
So that actually went nicely into this point that if you need to convert from a VDS to a project VCF, you can do so through a project called densification. So joint calling at scale. So moving back to this idea that if we had jointly processed gnomAD v4, these are the exomes, as a project VCF, it would have been about 800 terabytes. Using Hale's schema and their format, the entire gnomAD v4 exomes only takes up 18 terabytes. So this is an extremely scalable variant representation format that has very efficient storage. And this file is actually small enough that we can directly use it for QC rather than having to create a smaller intermediate file. So speaking of quality control, that's the next step in the pipeline, and there are two pieces in it: sample and variant quality control. So this is computational scientists in the production team, quality control or QC, 2 dimensions of the gnomAD release. And this plot on the left is just showing the growth from V2 in this little rectangle for the exomes to gnomAD v4. I'm sorry, I keep losing the pointer. And the same thing for the genomes. For quality control, I'll be focusing on the exomes, but I wanted to show both here just to show the scale of the issue. So starting with sample QC, this is a pipeline of all of the steps in sample QC. This is not actually designed to be readable. It's just to give you a sense of all of the things that go into this step. So this is the longest step of production. It is computationally very intensive, and the data heterogeneity is particularly challenging here. So the major steps of sample QC are hard filtering on the far left, platform inference, sex inference, relatedness inference, genetic ancestry inference, and sample outlier filtering. I don't have time to go through all of these steps, so I want to highlight some of the improvements that we've made to this final step, sample outlier filtering. And in this step, we compute per sample QC metrics, and then we filter samples that are outliers in that distribution. And to explain why we needed to make improvements, I actually need to roll back to gnomAD v3 quickly. So in gnomAD v3, we use this method where we regressed each QC metric with the principal components from our genetic ancestry group assignment, and then we plotted their residuals and determined outliers based on this distribution. So this is showing an example QC metric residual plot, and then these red dotted lines and the kind of red shaded areas are showing where we drew our cutoffs for outlier sample detection. If you zoom into the right side of the plot, though, you'll notice that there is a tail of samples that's getting filtered by our sample outliers pipeline. And if you look closely into these samples, you'll see that they're actually samples that are highly diverse from underrepresented groups that were introduced in gnomAD v3. And for more information about this particular filtering, I encourage you to read this preprint from Mary and Zan. But to put these samples into context, this is a plot of the PC1 versus PC2 from our genetic ancestry inference, and this is where those samples are falling in this space. So maintaining and increasing diversity in gnomAD is a top priority, so obviously we did not want to be inadvertently filtering these diverse samples. So to fix this, for gnomAD v4, we created a new outlier detection pipeline that was designed to be more inclusive of these diverse groups. So the first thing that we tried to look for was for each sample, we implemented a new method called the 50 nearest neighbors method, where we looked at the 50 nearest neighbors for each sample in Euclidean space based on the PCs. And then we determined outlier metrics based on those 50 nearest neighbors. So we plotted the distribution, and then we filtered based on 4 median absolute deviations from the median for here. So the orange dotted line is indicating where the sample falls in this distribution, and the red dotted lines are indicating where we actually drew the thresholds. We found that the 50 nearest neighbors approach alone still wasn't good enough, so we decided to move to an ensemble outlier filtering method. So that's this green color on the bottom. In this ensemble method, we did first the PC regression method on the left. So for the exomes, we actually stratified per exome capture platform, regressed the PCs, and then plotted the QC metric residuals. And then we also separately calculated the outlier distributions on the 50 nearest neighbors. We then only filtered samples that failed across both metrics. And this rescued both the diverse samples and also only filtered 1.2% of all of the samples. So now, after you determine the number of high quality samples that go into the release, you need to determine which set of variants are also high quality. So we assess variant quality in variant QC by evaluating a number of metrics, including de novo looking variants in trios, the transition to transversion ratio, proportion singletons, and many other things that I didn't have room for on this slide. We also specifically look at precision and recall in a pseudo diploid sample and NA12878, which is pictured on the right. And this plot is showing the precision and recall curve for the models that we trained for VQSR. So, sorry, for variant QC. For V2, we trained a random forest to determine a score of whether a variant was likely to be a true variant or an artifact. And in V2 and V3 and V4, we used GATK's allele-specific VQSR. So that's what this plot is showing here. Our team actually combs through all of these plots to draw cutoffs. So we filtered to keep 90% of all SNPs and 80% of all indels. And with this model for V4, we filtered 15.2% of SNPs and 29% of indels. So one thing that I will note is, because gnomAD is ingested into clinical labs worldwide, we really want to make sure that the samples and variants are high quality, but we do release all of our variants on the browser. So if you do want to see all of the variants, including filtered variants, simply toggle this checkbox on the gene pages. So if you scroll on the gene page down to the variant table, there's a little box here that you can toggle. And once you toggle it, the variant table will include all variants. So for example, this first line is showing a variant that did not pass our variant QC metrics. So speaking of the browser, the final step of our production workflow is releasing the data. So the production team here generates release files. And as part of this process, we generate all of the annotations that we need for release, including aggregate allele frequency information and in silico prediction scores. Once the data is ready, the software engineers and the browser team load all the data into the browser. One thing I wanted to mention here is that for the gnomAD v4 release, the production team handed off the data 2 days before we actually launched it. So just to give you a sense of what kind of timeline we're working on, we don't hold preliminary data. We release data as soon as we have it. And the browser team actually amazingly turned around and loaded it in 2 days. Simultaneously with the handoff and loading, the operations team, browser team, and production team work together to update all of the documentation on the website to make sure that it's reflecting all of the new data correctly. And finally, we release all of our data on Google Cloud, AWS, and Azure. And this data is available to download for anyone can download it at no cost to themselves. And this is a screenshot of the downloads page, which kind of explains how you can find all of our data sets. So to quickly summarize this section, the short variant production has 4 major steps. Each step requires a huge effort from 1 or more teams and extremely specialized knowledge. And then the entire process is possible thanks to this collaboration between the various teams. So moving into part 3, what is actually a gnomAD v4? gnomAD v4, as I've mentioned, contains over 800,000 individuals, and there are 2 data types, 730,000 exomes, so this includes samples from gnomAD v2 and about 400,000 samples from the UK Biobank, along with 76,000 genomes, which were previously released as gnomAD v3. This dataset is aligned to genome reference build 38, so if you are used to v2, that is aligned to 37, so this is a new change. Looking past the scale of v4 and into the diversity, you can see that we do have room to improve, but if you compare gnomAD v4 to gnomAD v2, on average, we're adding about 3 times more diverse individuals as compared to before. And the absolute number of samples with inferred non-European genetic ancestries is 169,000. And you'll see that this is larger than the entire v2 dataset. So the scale and diversity of v4 are enabling variant discovery, such that on average, we're seeing 2 variants every 3 base pairs in the exome. 42 million of the variants of v4 were not detected in previous gnomAD releases. And this is directly due to increased representation of diverse genetic ancestry groups. So to put it more simply, more diversity means more variants. In v4, we're actually seeing that we're nearly saturating discovery of highly mutable variant types. So this plot is showing the percentage of variants observed on the y-axis as a function of the number of individuals sequenced on the x-axis. And this is for 3 variant types, so synonymous, missense, and loss of function. So in v4, which is the very end of the plot here, we're observing 99.8% of all possible CPG substitutions and about 97% of all CPG missenses. However, we're only observing 83% of predicted loss of function variants. And if you expand to all variants, so this plot now includes non-CPGs in addition to CPGs, you can see that the variant discovery rates range from about 12% to 26%. So there's definitely room to grow, to sequence more samples and discover more variants. But to bring this back to v4, we wanted to look at variant discovery per genetic ancestry group. And specifically, we wanted to ask, how many variants are we discovering at or above an allele frequency threshold of 0.1%? And the reason we chose this threshold is because most variants at this threshold can be excluded as major drivers of Mendelian disease. So the more variants of this type that are in gnomAD, the fewer an analyst would have to look at in a rare disease patient. This plot on the right is showing the number of individuals in gnomAD colored by their inferred genetic ancestry group, and this is currently v2. The plot on the right now is showing all of the non-synonymous variants at or above a 0.1% allele frequency threshold contributed by each group. So you can see the total number for v2 is about 500,000 here. So what does this look like as you move from v2 to v4? This animation will play quickly, so I'll let it go a couple times. But so one thing that should stand out immediately is that the largest genetic ancestry group in v4 is European. But if you look at the variant bar on the right, the proportion of informative variants added by these samples has not increased despite the sample size increase from v2 to v4. And if you zoom in, the number of variants per sample is actually lowest for this ancestry group. So hopefully I have convinced you that more diversity means more variants. But why do we care about having more variants? Well, one of the reasons is something I've already alluded to. More variants means easier rare disease diagnosis. So most variants that cause rare disease are rare or absent from databases such as gnomAD. So the more variants in gnomAD that are at thresholds incompatible with disease, the fewer you have to look at in a patient, so decreasing your analysis burden. So to expand on the previous analysis, we wanted to ask a very specific question. How many variants would look like variants of unknown significance if you looked at their frequency in European groups only? And of those variants, how many would move to likely benign based on their frequencies in diverse groups, in non-European groups? So to show this visually, this plot is now showing the number of variants that would be classified as VUS using an even more stringent allele frequency threshold of 0.01%. But if you look at the frequency of these variants in these diverse groups, these variants would be considered likely benign. So in v2, there are about 420 of these variants. The increase in representation in V4 means that the number of these variants contributed has increased. So then more variants could be moving from VUS to likely benign based on this diversity increase. And that number is actually about 200,000. So to quickly summarize, more diversity means more variants. And more variants means that a rare disease diagnosis should be a little bit easier. And the scale and diversity of V4 means that more variants could be moving from VUS to likely benign. So up until this point, I've talked a lot about the variants that we've discussed that we found in gnomAD. But just as interesting are variants that we haven't yet discovered. So absence of variants in a data set such as gnomAD can imply intolerance or constraint. And to quickly orient you to what I mean by constraint, I'm showing two scenarios here. On the left is a gene that is relatively tolerant of mutation. And on the right is a gene that is relatively intolerant of mutation. So just based on baseline mutation and chance over time, mutations will arise in these genes. Some of the variants will not be tolerated and natural selection will remove them from the population. So for example, on the tolerated side here, you can see that most variants are tolerated, but individual 5's variant is not, and it has been removed. On the constrained side, you'll see that the opposite is true, that most variants are actually removed by natural selection. So then over time, a gene that is tolerant of variation will accumulate more variants as compared to a constrained gene, which will not accumulate quite as many variants. So when you aggregate across samples in a data set such as gnomAD, you'll see a reflection of this pattern in the variants found. So to quantify this in gnomAD, what we do is we compare the number of observed rare variants to the expected counts of variants generated by a mutational model that is built to be selection neutral. So the number of expected variants given neutral selection. And to visualize this, I'll be showing the number of rare variants in a gene on the y-axis and the expected counts on the x-axis. And this is what it looks like for synonymous variants in gnomAD v4. You can see that the observed and expected are highly correlated. The slope is about 1. And that means that we're observing basically as many synonymous variants as you would expect. And this aligns with our expectations, given that synonymous variants generally are not selected against. Also, because this is so tightly correlated here, we expect that our model is well calibrated for most genes. So moving into missense, you can see that there's a little bit more variation. The slope is still pretty close to 1, but there is a little bit more variation around this dotted line, which is what you would expect given the variable effects of this variant class. Finally, on the right are the predicted loss of function variants, and you can see that many more variants are falling beneath this dotted line, which means that we're not observing as many variants as you would expect. This, again, is what you would expect based on the deleterious nature of this variant class. So to focus on loss of function constraint, we know that most genes are actually depleted of loss of function variation. And many genes, including most haploinsufficient genes, are extremely depleted. So this plot on the right is showing the number of genes on the y-axis and the observed to expected ratio on the x-axis. And as you move from right to left, you're moving from less constrained against loss of function variation to more constrained against loss of function variation. And to put this into a single gene example, I've pulled out CHD7 here. Our model is predicting that we should see about 1355 expected synonymous rare variants, and we're observing 1337. So this is very close to 1. If you look at the loss of function variants, however, we expect to see almost 278 variants, and we're only observing 21. And this is what you would expect, as knocking out a single copy of this gene leads to an autosomal dominant congenital disease called Charge syndrome. So the gnomAD team, to try to make this observed to expected ratio a little bit more interpretable, the first score that we put out was the PLI score, or the probability of loss of function intolerance score. And the premise of this score is that you can group genes into 3 classes based on their sensitivity to loss of function variation. So the first class you can group genes into is null or completely tolerant of loss of function variants. So heterozygous and homozygous loss of function completely tolerated. The second category is recessive, where heterozygous variants are tolerated, but homozygous variants are not. And then the final category is haploinsufficient, where even heterozygous loss of function is not tolerated. The PLI score ranges from 0 to 1. with 1 indicating that a gene is highly constrained against loss of function variation. And this score is actually very dichotomous. So most genes either have a score less than 0.1 down here or greater than 0.9 up here. And there's very few in the middle. And to bring this back to the gene example for CHD7, the PLI is 1, which again is what you would expect. So to improve on the dichotomous nature of the PLI score, the next score that the gnomAD team put out is the loss of function observed to expected upper bound fraction. It's a mouthful, but it's the LUF score. And this is a continuous metric. So this directly measures biological significance. You can pull the LUF score at any point in this spectrum, rather than having to say, like, is it only on this side or only on this side? And because this is using the upper bound of the confidence interval, this is correcting for gene size a little bit better. So we know that the number of observed rare variants in a gene is correlated with the size of the gene. The LUF score also ranges from 0 to 1, but you'll see that it's in the opposite direction of PLI. So a PLI of 1 is indicating that a gene is very constrained against loss of function variation, but a LUF score of 1 is indicating that a gene is not constrained. And finally, to bring this back into the gene example one more time, the PLI for this CHD7 is 1, but the LUF is 0.11. So we recalculated the LUF score on gnomAD before and applied it to 3 gene lists. This is again, right to left is moving less to more constrained. This is the LUF decile on the x-axis and the percentage of the gene list on the y-axis. You can see that for the most constrained LUF deciles, they're enriched for haploinsufficient genes. The least constrained LUF deciles are enriched for olfactory genes, and autosomal recessive genes kind of span this middle section. This is what you would expect given the function of each of these gene lists, and this is the pattern that we saw in gnomAD v2 as well. So moving on to an application of LUF. So one major use case of this score is to identify enriched variant classes in rare disease. So this plot is now showing the rate ratio of de novo predicted loss of functions in developmental disorder cases versus controls. And again, right to left is less to more constrained. So this is LUF decile on the x-axis. There are 2 bars on this plot. So the pinkish reddish bar is indicating the LUF score from V2, and this kind of teal color is the LUF score for V4. You can see that the most constrained LUF bin is showing a strong enrichment of de novo protein loss, predicted loss of functions in cases versus controls. And if you move from V2 to V4, the V4 LUF is actually showing an even stronger enrichment, so an even stronger signal here. So the scale and diversity of V4 are increasing our resolution of the LUF score here, which is also increasing our understanding of kind of the genome function. If you're interested in learning more about constraint, I do encourage you to navigate to the gene pages. So constraint is available on this table on every gene page, and our tables are also available for download on our downloads page. So just to quickly summarize, gnomAD v4 is available now. We've added about 169,000 individuals of inferred non-European genetic ancestry. Excuse me. We've added gene constraint on genome reference build 38 for the first time. We've also added filtering allele frequency to further enable clinical variant classification. So this is jointly calculated across the exomes and the genomes. And for the first time, we're thrilled to announce that we've released structural variants and copy number variants simultaneously with our short variants. The V4 release, the 4.0 release, is a minimum viable product release. So that means that features available in previous releases are not yet available, but will be coming soon. So I encourage you to check back in the future. And finally, I'd like to wrap up with answering a few of the common questions that we received. So the first one that we get pretty often is, does gnomAD only have healthy individuals? And the short answer is no. So we remove cohorts where possible that we know that they were recruited for pediatric disease, but we can't ensure that no individual with the disease isn't present in gnomAD. However, we don't expect the incidence of disease within gnomAD to be at a frequency higher than what you would expect. We expect it to be equivalent or lower than what you would expect across the general population. And this is especially true as we start to include more biobank samples. We cannot ensure that every sample in gnomAD is healthy. Will you release V4 on genome reference build 37, or will you support V4 on 37? No, we are encouraging all users to move to 38. And if you do need to lift over, if you go to the V2 location, there should be a liftover link that will click if you click that link, it'll take you to the lifted over location in V4. So what features are not yet available in V4? So we have a help and FAQ page section about this, but previous scores, like the PEC score or multinucleotide variant calls, oh, sorry, that's a typo, variant co-occurrence, all of these are not available yet in V4, and they will be coming soon. We do list on this page where you can find the current versions of these metrics, so I encourage you to check that out. And again, please do keep an eye out on this space. If you have other questions, we do encourage you to check our help page where we have quite extensive documentation, or check out our new forum where we're encouraging all of our users to ask and help each other with gnomAD related topics. So with that, again, gnomAD is only possible thanks to a wide network of individuals. So I'd like to thank everyone in the project, particularly the core team pictured on the left and the individuals who contributed data. I'm happy to take any questions.
Thank you so much for a really wonderful and clear talk. And I will just say, I really liked the idea of having an FAQ at the end of a talk. That is an amazing feature. And maybe many of the questions that would have come up, you've already addressed. I will also welcome anyone here to come to the microphone with a question. But I will start out by reading one that appeared in the online Q&A. Thank you for an informative talk. Is more information on how splice AI annotations were generated going to be released? They're not in the default output format. And I checked if you couldn't line up the max delta with results for the variant on the lookup tool.
Oh, I see. Okay. So we didn't actually generate the splice AI annotations. The Luminum team annotated those for us on the data set. So I would encourage you to reach out to their team if you have questions. But one thing I will note is that on the gnomAD Hail table page specifically, we mentioned that for splice AI, we tried to pick the maximum score for main select transcripts. So we prioritized scores for main select transcripts where possible. Canonical transcripts were not. And that is the score that we're displaying. So it's, if that answers the question of why that doesn't match up. Oh, we also encourage people to use the splice AI lookup tool if they have more information and want to dig up more details about the score.
It certainly seems to answer the question. If the person who posted has further questions, please post those here. Any questions from the room? I will raise then one just purely out of curiosity. It made sense and was exciting to see that the olfactory genes indeed come out on the far right there. I wonder if the database is now large enough, or you anticipate at some point in the future it being large enough that you'll be able to, with confidence, detect some things for which the number of variants is way greater than you'd expect in a way that could suggest other forms of selection on gene types that we haven't yet anticipated from first principles.
Yeah, that's a really interesting question. So for constraint, we haven't historically focused on that. So we've focused more on depletion of constraint because that's kind of how we're aiming to for people to use this score. It's really best powered for genes that are severely selected against that really impact reproductive fitness. So we haven't really calibrated it to look at the other end of the spectrum, but it's something that we can consider.
Thank you.
Yes, hi, Ingrid Christopherson. I was wondering also about the constraint metrics. Have you also calculated that on like exon level or domains of the genes?
So this is gene level. This is just across the gene. So this is for synonymous, missense, and loss of function across the gene. In the future, we are hoping to release regional missense constraints. So that is one of those features that isn't included in the minimum viable product release, but is currently available on V2. But that is only for missense variants, and that is at a base level resolution.
Thank you so much. I was wondering, first, sorry, there was one. Do you mind going back to the page that has the community link, community forum? Thank you. There was just a request from someone online. And then I was very interested in your choice of 50 when you were choosing the outliers, because on the scale of the total number of samples you were considering, that seems like a relatively small number. And I guess I have two questions. One is the technical, like how did you arrive at 50? And the other one's kind of more big picture is what do you think that means about the kind of the uncaptured human variants and the kind of need to, the ultimate scale of database you would need to perhaps not have outliers based on ancestry anymore.
Oh, okay. So for the first question, 50, I think we actually tried a few different thresholds, but I'm not 100% sure at this point, but I believe we tried 50, I think 100 and a couple of different numbers, and we kind of settled on this as like the best balance of compute time and information gathered from the other samples. As for your second question for how large the data set is, I don't know if there's a size that where you would be able to stop detecting outliers based on ancestry. I think it would be more the sample composition, and I don't know what that would look like. You would have to have much more equal representation and include more groups that are just not represented in databases like gnomAD right now.
Great. Well, thank you so much for a lovely talk today. And then if anyone has questions, feel free to come up, and otherwise we'll reconvene in about 20 minutes for the main session.